I'm Dr. Alice Pyne, and I'm a lecturer in soft matter in the Department of Materials Science at the University of Sheffield. And my research focuses on using high-resolution atomic force microscopy to look at the structure and interactions of DNA. And part of the reason for that is a few years ago we were able to be some of the first researchers to see the double helix of DNA on a single molecule in liquid. And what we were able to see was when we looked at this single molecule, we were able to see that the structure of DNA was more variable than we thought it might be. So we still saw this incredible double helical structure, but it had variations within it. Some of the coils were bigger, some of them were smaller. And so I've spent the last few years of my research trying to understand where those variations in structure come from and what they might mean. So trying to understand how DNA as a molecule is more different and variable than we thought it was, and how that affects how it interacts with other proteins, other DNAs, how it folds up, and how all of that works together to make the incredible DNA within your body compacted down into each of your cells. So in each of your cells within your body, there's 2 meters of DNA roughly, and that all has to be compacted down into the nucleus, but then read out and used throughout its lifetime. When we think of DNA, we think of it just being this short helix, but actually, if you've got 2 meters of DNA bundled into a small space, you have to work out how it bundles itself, how it arranges itself, and how it does that in a way that still means it works. So what we're able to do is look at the individual pieces of DNA on a microscope, both to look at their structure, but then also to examine them interacting with other things, to understand a little bit more about how your body works. AFM, or Atomic Force Microscopy, is a really incredible technique for looking at DNA, partly because you can work in liquid. So all biomolecules are found naturally occurring in water or liquid of some sort. But a lot of the techniques we use to investigate them aren't able to work in liquid. They work in vacuum, they work at low temperatures, they work in crystal structures. So we have this huge advantage that we're able to work in liquid. And the advantage that gives us is that in addition to looking at structure of molecules, we're able to look at dynamics. So it's brilliant that we're able to see these molecules. I still find it incredible. But one of the things we want to do with that is actually get numbers and statistics for how these molecules appear, what form they take, what structure they're in, and how many interactions we see. So we've written some Python code that enables us to analyze thousands and thousands of images in minutes and generate statistics on thousands of molecules and understand how conformational changes occur, how these images actually relate to biophysical interactions, interrogate binding efficiencies, KDs, all of these important things that we need to know for understanding how proteins bind and also how that might be affected by therapeutics. So a lot of the drugs we take, from antibiotics to anti-cancer drugs, target proteins that interact directly with DNA. And we've been using those for years without actually understanding how they work. And although that's not the case now, we know how most of these things work, there's still a lot we don't understand about their exact mechanism of action, how effective they are, and why things don't always happen as we think they will. So in the case of CRISPR-Cas, one of the real discoveries of the last decade, it's incredibly effective, but there's a lot we don't understand about why it doesn't always work, or sometimes it works when it shouldn't work. And so part of what we're trying to do is say, okay, great, we've got these drugs and they work against this protein, but how do they work? What part of the DNA protein interaction cycle do they target? How can we make that more effective? Actually, when we start putting it into the more complex environment of a cell, or even just interacting with DNA or in liquid, it actually can behave quite differently to how we expected. So one of the things we're trying to do is interface our materials science biomolecular research with the medical, the therapeutic industry. In my group and in my career, I want to have a long-lasting effect on the development of new therapeutics. So we desperately need better anti-cancer drugs, more antibiotics, a better understanding of how they work, and new avenues to develop them.